webinar. So welcome everyone to the Baruch Blumberg Institute's webinar. Today we have invited Dr. Barbara Rehermann to present some of her recent studies on the liver-resident immune cell in hepatitis B and hepatitis D. So we all know Barbara for a long time, that almost in the last 3 decades, she have made a tremendous contribution to our understanding of the host immune response against hepatitis virus infection. And especially, in recent, I was caught by one of her paper published in Science Translational Medicine on the interferon beta treatment efficacy studies against hepatitis B. So, working in the hepatitis B, we all know that the only pegylated interferon that was actually can cure a small fraction of the chronic hepatitis B infection patients. So, but for a long time, people don't know why someone that treatment works, someone not. And Barbara's work that give a very nice, interpretation of this long-standing clinic dilemma. So today, let's welcome Barbara to her presentation.
Thank you very much, and good morning. I hope this is the last time we have to do it via Zoom. We are really happy to soon travel again, and this will be much more personal. I will try to stay within time, so about 40-45 minutes, and present some recent findings that were quite surprising to us, and may end up with some novel hypotheses that I hope will trigger more research and clinical studies. So the title of this presentation is Liver-Resident Immune Cells in Hepatitis B and D. And I will present basically 2 studies that were just completed, starting with hepatitis D. So why the focus on liver-resident immune cells? So conceptually, we are always thinking about liver-infiltrating immune cells in hepatitis. And that is certainly the case in acute hepatitis, as we have learned from mouse models, from Frank Cesari's studies, that if you inject immune cells, CD8 cells, with the HBV receptor into transgenic mice, then they go into the liver and start an inflammatory infiltrate that basically results in lysis of hepatocytes and ALT increase, acute hepatitis. But what is going on in chronic hepatitis? That is really not as well defined. Studies from Marla Mainy's group, Laura Pallet, have shown that there are resident immune cells, and our recent studies also focused on this. So I'm going to talk about T cells in the liver. As you see here, they can touch through the open liver endothelium cells, through the LSAC, HBV infected and epitope expressing cells, and then infiltrate the liver, get activated. I will talk about Kupffer cells and also, of course, innate immune cells, NK cells and mate cells. So starting with hepatitis D. So we have a study here at the NIH in the liver disease branch run by Chris Coe and Theo Heller, trying to understand HDV pathogenesis and evaluating new treatments. So we have received liver biopsies and blood lymphocytes from these patients, and we try to understand why this very small virus has such a large impact on the HDV pathogenesis, basically causing an infected liver to turn into a cirrhotic liver at a much accelerated rate, much faster than you would see with hepatitis C and hepatitis B. And so what is known already is that there's a large liver immune cell infiltrate in hepatitis D virus infection, And the first analysis that we did here shows that, of course, liver enzymes are much higher in HDV, HBV co-infected patients than in patients with HBV monoinfection. And that correlates with the inflammatory score in the liver. We found many years ago already, that's not yet published, that serum chemokines are much higher in the co-infected patients than in the patients with just chronic HBV monoinfection. And this is especially true for the CXCR3 chemokines CXCL9 and CXCL10, and not for the other ones here in our study. You can see the individual data points on the next slide. So you see here that CXCL9 levels correlate very nicely with liver ALT activity and also with histologic activity index in the liver biopsies. And this is not the case for the other ones that we examined here. So we have also shown in still unpublished studies that if you infect hepatoma cells, HUH7 cells with HDV, and transfect them with the plasmids and then harvest the produced HDV and then infect primary human hepatocytes, that this results in HDV production in the cells, but also interferon beta induction. So that's a type 1 cytokine response. And this results in the chemokine expression here at the mRNA level and also at the protein level. And you can block it if you add the vaccinia virus B18 protein, which blocks the interferon response. So HDV infection, as also shown by others, by Maura Dandri in mouse models, induces type 1 interferon responses. And this, of course, activates immune cells. In addition, we have also found in a recent study here in the plasma of the infected patients that there's quite an increase in IL-12 levels. And both the P40 and the P70 subunits is increased if you perform ELISAs on the plasma samples. Likely, there's also an increase of other cytokines, such as IL-18, IL-15, but these need to be studied in the liver, which we have not done because, for example, IL-15 is transpresented. So it never really leaves the immunological synapse. It binds to the IL-15 receptor on the cells that produce it and then directly stimulates the neighboring immune cells. So plasma samples would not be adequate to really assess IL-15 or even IL-18 levels. But so there's an increase in type 1 interferon and also in other pro-inflammatory cytokines that are known to activate immune cells. And so not surprisingly, when we analyzed paired blood and liver samples from HDV-infected patients and compared them to those of uninfected controls, then these are not patients, these are is liver resected material from liver resections that we received. Then you see that the NK cells are activated, express HLA-DR more in the liver than in the blood, and there's a higher activation status in the liver of HDV-infected patients than in non-HDV-infected control tissue. And the activation status of the liver NK cells correlates inversely with HDV RNA, suggesting that this is really associated with an antiviral response. But what about T cells? So that was our main goal, to analyze T cells in the liver. And at the time we started these studies in about 2017, I think, there was not much known about T cell responses. Epitopes in HDV infection had really not been identified. And then in 2019, our study came out at the same time as studies from the University of Freiburg that also published CD8 Tcell epitopes. And we have done it here the classical way. We started with large overlapping peptides, overlapping the entire HDV protein, identified large peptides that were recognized by CD8 T cells, and then went down and synthesized truncated peptides and tried to map the minimal optimal epitope, which is here shown in a dose titration curve in red. So this is the peptide that has the strongest CD8 T cell stimulatory capacity at low concentrations. These studies were started basically when Helene Kefalakis joined the lab. She worked on HDV here in the immunology section, and she just recently completed her postdoctoral fellowship and is now joining the Medizinische Hochschule Hannover in Germany with Heiner Wedemeyer. So coincidentally, the place where I originally came from, which is very nice to see, we came full circle. So, but what did we do? So we mapped all of the CD8 T cell epitope in order to synthesize multimers that can stain HDV-specific T cells without changing their function. So any T cell that has the appropriate T cell receptor and recognizes the peptide in the context of the patient's HLA molecule will stain, as you can see here in the FACS plot. And there are many commercially available reagents now. If you know the minimal optimal epitope, we usually try to use dextramers, as you can see on the left. They cross-link many different T cell receptors and so have a very tight binding. You can also use pentamers or tetramers and different valencies depending on the reagent that you use. And what we have identified then by flow cytometry are CD8 T cells that recognize all of these individual peptides. And out of curiosity, we were very surprised to find that most of them are presented by HLA-B alleles. And this is the same what Christoph Neumann-Hebelin from University of Freiburg found in independent studies. For some reason, there are many more immunogenic epitopes presented by HLA-B alleles than by HLA-A alleles. And we both speculate that HDV has already adapted very much to the HLA-A alleles that are dominant worldwide, and so does not induce a strong immune response anymore, and that it's much more immunogenic in the context of HLA-B alleles. But these are just hypotheses. At present, we don't really know why this is the case. It may be important for vaccine development for a variety of infectious diseases, that it might be better to use epitopes that are presented by B alleles. So with all of these epitopes, we found many T cell responses. We characterized them in this publication that is shown in the right lower corner. But we also found that at least half of the cages had HDV escape mutants. So when Helene sequenced the virus that was infecting and present in the blood of these patients, the infecting virus sequence did not have the prototype epitope sequence, but had a sequence with a different amino acids, as you can see here in the second sequence on the top. And these were not recognized. These sequences in the serum of the patient were not recognized by the CD8 T cell responses that stained so well with the prototype epitope sequence. So as we know from HCV infection, the same happens here. You have viral escape mutants and the virus escapes from the T cell response, which may be one of the reasons why the infection is typically chronic. And this was shown here with interferon gamma production of CD8 T cells as a readout. And we, which means I, also did the classical cytotoxicity assays in the same way I had learned it as a postdoc in Frank Cesari's laboratory. These are standard chromium release assays. And you see that the prototype sequence on the right is recognized and results in lysis of the target cells that are pulsed with the peptide. But the autologous sequence of the patient is much less recognized or not recognized at all. So it's effective escape from cytotoxic T cells. So then, if this is the case, what really happens in the liver? What drives the liver injury? So up to now, all of the T cell analyses were done with peripheral blood samples. And we had the good opportunity to obtain small pieces of liver biopsies from the clinical studies from patients who were followed before and during treatment. And we had the opportunity to stain both liver samples and blood lymphocytes in parallel in the same assay. So what about the intrahepatic immune responses? So when we use the same dextramers and pentamers that were matched to the epitopes that the patients recognized and the HLA that presented the epitopes in each individual patient, and then stained blood and liver samples in parallel in the same assay, fresh, the same day we received the liver biopsies, you see on the left graph that there's a much higher frequency of HDV-specific CD8 Tcells in the liver for any given epitope than in the blood. And that's not surprising because we had expected the CD8 Tcells that recognize HDV to basically be enriched in the liver. But what is still surprising to me is that the frequency of these HDV-specific CD8 Tcells is quite high. So for any given epitope, you have about 6% to even 10% or above 10% of the liver-infiltrating CD8 Tcells recognize that specific peptide. So even though HDV is small and encodes a small protein or peptide, it must contain multiple CD8 Tcell epitopes, so you can easily assume that 20%, maybe 25% of the CD8 cells in the liver are specific for HDV. And then with this analysis, we basically combined our immunophenotyping data from liver and blood samples and analyzed them with phenograph clustering using this Flitzny approach that is shown on the right. And this approach, combining liver and blood samples, the data from both samples, and combining them and analyzing them in one set, had the advantage that we did not need to downsample the analyzed events. And therefore, we can analyze both low abundancy HDV-specific cells and the total CD8 Tcell fraction in parallel at the same time. And as you can see here, we had overall 25 clusters, which are numbered in the middle graph. And each graph shows blood and liver samples. And these clusters formed basically 3 different islands: naive cells, memory cells, and TEMRA cells. And if you look on the right graph, on the liver specific, the immune cells in the liver, then they basically cluster in the upper island, which are TEMRA cells, and then also in the right island, separate island, which are memory cells. So the red clusters of red spots are in the upper island and in the right island. And the lower left island, basically naive cells, as you can see in the middle graph, are only found in the blood and not in the liver. So the cells have a differentiated phenotype in the liver. And then when we look at all of the individual clusters and compare them in blood and liver, and so I have to say each liver sample were about 4,600 events median, and the blood samples were about 1 million events. Then you can see all of the different clusters here, naive cells, memory cells, and TEMRA cells. And on the right, you see the frequency of cells, events in each cluster. So in the blood, the most frequent clusters, the most abundant clusters were 1 and 2, with about 15 and 14% of the blood CD8 cells falling in this category. And in the liver on the right, you see clusters 17, 3, and 8 that contained a lot of cells, combined more than 70% of the cells. And you see at the bottom, the beauty of this analysis is that you can also assess the HDV-specific cells, as you can see on the left, which are marked as an HDV-specific T-cell receptor. So these are the ones that stain with the multimer presenting the HDV peptide by the correct HLA molecule. And you see that these cluster in different groups in the blood and in the liver. So this was this unbiased analysis that we took. Rather than gating individually, it was a phenograph analysis here and unbiased analysis of these clusters. So HDV-specific T cells were, for example, found in cluster 14 in both in the blood and in the liver. But when we then compared the phenotype of this cluster, there was much higher CD39 and NKG2D expression in the liver HDV-specific cells than in the blood. Overall, the most surprising for me personally was that all of the activated CD8 T cells that express the marker CD38 were basically in the liver resident population. So you see here the second population from the left. These are CD69 positive and CXCR6 positive, and they make up almost all of the activated CD38 positive HDV specific cells. So this means that all of these HDV specific cells have a liver resident phenotype because CD69 and CXCR3 expression basically characterizes the cells that are resident in the liver. And if you use a different readout, if you look at CD107A expression, that's a degranulation marker, so it indicates degranulation and is a biomarker for cytotoxicity. Then you see that the greatest percentage of the CD107A expressing degranulating cells is also contained in the liver resident population. So again, CD69 positive and CXCR6 positive. So really, these cells are resident in the liver, very different from the cells in the blood, indicating that we really need to look in the liver to characterize fully the HDV-specific immune response. And we don't know when these cells have seeded in the liver. It could have been a long time ago when the infection was acute, or there could be slow renewing potential of these cells in the liver, or there could be a very small continuous influx from the blood. So that's really not known. I think we need mouse models to study that further. And very interesting, if you look at all CD8 T cells, so not just the HDV-specific ones, but at the total CD8 T cell population in the liver, you see exactly the same thing. Most of the activated total CD8 T cells are liver resident, and also most of the degranulating CD107A positive total CD8 T cells are also resident in the liver. So it really makes us wonder what can be learned from just analysis of blood samples, which of course is very difficult for immunological treatment, immunotherapy, because we don't have access frequently to liver biopsies. So we did multiple comparisons. So we compared blood and liver CD8 T cells, and you see here blood on the left, liver on the right, and all of the different markers that we studied on HDV-specific cells and total CD8 T cells. And what is apparent, I think, already from this radar plot analysis is that there's not so much difference between HDV-specific and total CD8 T cell phenotype. So on the left, you see the biggest difference between basically the blue curve and the red outline in the middle is for CD69 and CXCR6, which, as I had shown before, are the liver resident markers. But otherwise, there's not much difference. Statistical significance is reached for NKG2D, but that's not really well visible here because the lines are overlaying. And if you look on the right and compare basically the blood and the liver radar plots, then you see that there's a much bigger difference between radar plots in blood and liver. So the environment determines the phenotype than the difference between HDV specific and total in each compartment. So we think that much of the phenotype of these liver-resident T cells is indeed driven by the specific environment in the liver, and that we see similar phenotypes for both HDV-specific and total CD8 T cells. So this raises a question, how these cells get activated in the liver. If there was a difference between HDV-specific and HDV-specific and total, then basically we should also see a difference with escape mutations, because this would determine whether the T cells still recognize the viral sequence or not. But here, when we stratify by HDV escape mutation, so the open circles are CD8 T cells that cannot recognize the predominant HDV sequence, whereas the filled circles are the ones that recognize the sequence, where there's no HDV mutation, then you basically see that there's not much difference. Each of these individual groups contains both T cells that cannot recognize the HDV sequence, open circles, and those that can recognize the sequence, closed circles. And there's no real stratification, there's no big difference. So they basically cluster together. The only big difference is here the significance comparing these two green areas. So comparing the frequency of HDV-specific T cells that express PD-1 and CD38, so very activated, almost exhausted phenotype in blood and liver, you see a much higher frequency in the liver on the right than in the blood on the left. But it suggests that this is not really driven by the HDV-specific sequence, because there's no difference between filled and open circles. If you look at other phenotypes, for example, PD-1 and CD217 expression, then it's the same. So you see open circles and filled circles cluster together. There's not much difference. But if you compare, for example, the yellow shaded areas in blood and liver, you see a much higher frequency of PD-1 negative, CD127 positive, so classical memory cells in the blood, than in the liver. And in contrast, in the liver, if you compare the blue areas, then many more of the cells are PD-1 positive and CD127 negative, so just the opposite than in the liver than in the blood. So again, it suggests that the liver environment determines the phenotype of the cells and not so much whether they recognize a specific HDV sequence or not. So what does that tell us about correlation with clinical parameters? We see here, as expected, when we look at HDV-specific CD8 T cells, for example, the terminally differentiated ones that express CD57 in the upper graph, or correlations with HDV RNA, liver inflammation, and AST values, you see correlations between the frequency of those HDV-specific cells and the individual clinical parameters. But if you do exactly the same analysis with total CD8 T cells, you get the same kind of correlation. So in both cases, you see significant correlations, irrespective of whether the T cells are HDV-specific and recognize their target or not. So this results then in the hypothesis that much of this immune cell activation in the liver is not driven by recognition of HDV peptides, but instead driven by inflammatory cytokines in a bystander manner. And we know that this can happen. So these are not HDV samples, but these are samples from healthy blood donors that have been stimulated with IL-15, for example. You see that with IL-15 stimulation, you can drive NKG2D expression, which is one of the markers we have observed in the liver on HDV-specific and total CD8 T cells. You can significantly increase that expression. And if you increase NKG2D expression, then you can induce cytotoxicity against hepatoma cells, as you see in the middle graph. And if you block NKG2D with an antibody, then you can abrogate the cytotoxicity. So there's a case to be made for cytokine-driven T-cell activation that results in cytotoxicity and lysis of hepatoma cells. And very interestingly, HDV infection drives the expression of the ligands for this NKG2D. So the ligands are MIC-AB and the ULBP proteins. And if you compare here hepatoma cells that express or don't express HDV, and these are cells that we obtained from Stefan Urban, who kindly provided these, then you see that the HDV positive cells have much higher MIC-AB expression and much higher ULBP1 expression and ULBP2, 5, and 6 expression than the HDV negative ones. So also HDV itself can drive this supposedly cytokine-induced lysis of hepatoma cells that contributes to an inflammatory phenotype in the liver and supposedly to disease progression. So that is the hypothesis that we have now at the end of the study. And I would expand that. I would say this is not unique for HDV infection. This is also present in HBV infection, as shown by Malamaini, Laura Palais, and probably also to a lesser extent in HCV expression in infection, because many of the cytokines that we observe in HDV are also present in HCV. So this basically is the first part of my presentation on HDV. So then going on to treatment responses, and this is a study I would like to summarize briefly on HBV responses that were mentioned in the introduction. So for HBV, we are all looking for new treatment modalities that basically induce functional cure, which is defined as absence of HBs antigen in the presence or in the absence of anti-HBs antibodies. It can occur spontaneously in chronically infected HBV patients, but only at a very small conversion rate each year in chronic infection. So it's not a frequent event, but it can occur spontaneously, which triggers hope that we can induce the immune response to also do that during treatment. And so far, the only terminal treatment that can be stopped and can result in HBs antigen loss is still PEG interferon alpha treatment. And you see here that if you suppress HBV replication with tenofovir and then add PEG interferon alpha treatment for 48 weeks, this results in the highest rate of HBs antigen loss, as shown in this study with really a very large number of patients and comparing all of these different treatment regimens. But overall, we have to say that even if you treat for 48 weeks, only a small percentage of patients loses S antigen, and this occurs with a delay. It doesn't occur immediately, as we all know. It happens sometimes long after interferon treatment was stopped. So we assume that these are immunological events that are triggered, but we don't really know what these are. And so in a study that we did here at the NIH with Mark Garney, my clinical collaborator, we were able to study patients who had received nucleoside analogs for more than 4 years. And they were treated then with PEG interferon alpha add-on therapy for 24 weeks, and then followed for 2 years after that, followed for 4 years after that. And most of these patients were HBe antigen negative, so it was really not possible to look at loss as E antigen as a readout, and it was also not possible to look at HBV DNA level decrease, because these patients were already suppressed and had no detectable HBV DNA for more than 4 years. So this made it harder, but it also made the baseline cleaner, because we were able to detect very unique immunological events. And this is what I'm going to describe. So how do we want to monitor the immune response to PEG interferon alpha injection? So of course, we can monitor this with liver biopsies and looking for IgG expression level. And these IgGs include certainly IgGs, proteins that interfere with HBV replication. But this is difficult because we don't have so many liver biopsies and we don't have them prospectively during treatment. So another way to assess the immune response to the injected PEG interferon alpha is to just look at blood samples and to measure the immune response of NK cells. So NK cells are frequent in the blood, which makes the flow cytometry readout very easy and straightforward. NK cells respond very rapidly to interferon, and they secrete, as one of the readouts, TRAIL. And TRAIL binds to TRAIL receptors on hepatocytes and can trigger apoptosis. So TRAIL is not just an IgG itself and an easy readout. It's also an effector molecule that plays a role in the immune response in the liver. So we just decided, based on studies that we had done in HCV infection, to monitor the NK cell response as a first readout and to just assess TRAIL as basically an indicator IgG and an indicator of whether immune cells get activated and whether there are differences in the patient immune response. So this study was started by Fabian Bolte, who was then a postdoc in the lab. He analyzed the first samples and then was rapidly joined by Akira Nishio. Both have also moved on so far. Fabian Bolte returned to Germany, but is now at University of Virginia in a clinical residency, and Akira Nishio just recently returned to Osaka University in Japan. So, and very interestingly, in this paper here, where we describe the results, we found a huge difference. About half of the patients had a very strong NK cell response, easily detectable, with up to 40 to 80% of the NK cells expressing TRAIL 6 hours after the first PEG interferon alpha injection. And then the other half of the patient had almost no detectable response 6 hours after the PEG interferon alpha injection. So there was a rapid response in about half of the patients, and at the most maybe a delayed response, but not detectable within 6 hours in the other half of the patients. And we were lucky because this was a pilot study. We had very few patients enrolled in this clinical trial, but we had, as it's typical for NIH, we had a very close follow-up with liver biopsies before treatment at 0 hours and 6 hours, blood samples at both time points, and then at very frequent intervals thereafter. And this made this analysis possible. So when we look, for example, at the full change in the TRAIL expression level on the CD56 bright NK cells, which respond mostly to interferon with this very strong TRAIL expression, then this correlates nicely with the peak ALT level that we observed at these very frequent follow-up periods after PEG interferon injection. And when we look long-term at HBs antigen decline, we also saw a significant difference between these groups. So just almost reaches significance, it's 0.051 p-value in HBs antigen decline at the last follow-up time point. But again, I have to say this is a very tough readout because we were not able to look at E antigen loss or E antigen serum decline or HBV DNA decline in this cohort because it was already HBV DNA negative and E antigen negative at the start of the study. So really S antigen was the only clinical readout that we could have here. So the question was the correlation with this ALT increase, AST increase, whether TRAIL induction on the NK cells causes hepatocyte death. And we were able to observe the same TRAIL induction on liver NK cells at the 6-hour biopsy and on blood NK cells. And in the staining of liver biopsies at the 6-hour time point, you see here that there is expression of TRAIL receptor 1 at the same location as there's expression of cleaved caspase 8, which basically indicates a cytotoxic T cell, which indicates a cytotoxic response and apoptosis of these hepatocytes. And merged, these two images overlap almost perfectly. And you see that patients with the NK cell response on the right had also higher peak plasma concentrations of caspase cleaved keratin 18 and also had higher peak serum HBV DNA levels. And this was also surprising because where does this HBV DNA come from? As I mentioned, these patients were on nucleoside analog treatment and had undetectable HBV DNA before and throughout the studies. So we hypothesize that this peak in serum HBV DNA comes from basically from lysed hepatocytes that release HBV DNA, which is then detectable in the blood. And again, this is an indication that there's an NK cell effector response in the liver. So going back to a broader assessment of IgG expression and inflammatory cytokine responses. So up to now, we had just used TRAIL, but what happens if we monitor the PBMCs for a broader IgG response? So you see here that patients with NK cell responses had more IgGs increasing 6 hours after PEG interferon alpha injection than those without. And the fold induction of the IgGs is also greater in patients with NK cell response than those without. So this is on PBMCs, IgG expression in PBMCs as assessed by nanostring analysis. But Mark Garni also has liver biopsies, and he has seen the same in the liver biopsies when he measures IgG expression levels there. So why does half of the patients respond in a strong way to the injected interferon with an IgG and NK cell response, and the other half doesn't? Is there a difference in their general ability to respond to PEG interferon? So we did then interferon signaling analysis and confirmed that interferon signaling is indeed intact in both patient groups. So this was done by taking PBMCs prior to treatment from both patient groups. So they had not been exposed to any PEG interferon, but we took the PBMCs prior to treatment and then exposed them to interferon alpha and to interferon beta in vitro, and then performed analysis to look at STAT1 and pSTAT and STAT2 phosphorylation, and we saw basically no difference. So the ability of the patients and the PBMCs to respond to interferon was not different. So what then contributes to this differential interferon response and differential NK cell activation? So very surprisingly, again, we measured PEG interferon levels. We performed an ELISA for PEG, and we also performed an ELISA for interferon alpha in the blood. And here's just a PEG interferon ELISA result shown. And you see that there are two different patient groups. So half of the patients 6 hours after the injected PEG interferon alpha had quite high PEG levels, and the other half had significantly lower levels. And the same difference was also seen 24 hours after PEG interferon alpha injection. And then in the end, both patient groups reached the plateau. And interestingly, I've marked this again in pink and in light blue. The pink patients are the ones with an NK cell, with an NK cell response. So if there is a higher level of PEG interferon in the blood, then there is an NK cell response. And if there are lower levels, there's no NK cell response. So there was one patient also who had very high anti-PEG IgM titers at the beginning of treatment. And what we saw is that the interferon alpha level and in parallel also the PEG level in the ELISA increased only when the anti-PEG IgM titer decreased. And so this suggested there's an interaction between antibodies to PEG and the PEG interferon alpha that's injected. And of course, there are two processes that are going on. You have a production and a stimulation of antibody production, and at the same time you have removal of antibodies and PEG from the blood. And basically the balance of these two processes determines how much PEG interferon you have in the blood at these early time points. So there's a plateau at week 2 and week 4, as you can see on the left graph, but I would like to mention that the samples at week 2 and week 4 are always drawn 1 week after the last PEG interferon alpha injection. So only at the very first time point do we have samples that are drawn 6 hours and 24 hours later. So it's very well possible that even after the second PEG interferon alpha injection and even after the fourth PEG interferon alpha injection, that you have the same kinetics that after the first one, with the difference between both groups. But then if you test 1 week later, basically just prior to injecting the next PEG interferon alpha dose, then there's no difference anymore. There's a plateau. So how do the antibodies influence the PEG interferon levels? So there are already papers described, mostly in animal models, that describe the ABC phenomenon. That's accelerated blood clearance of PEG conjugated proteins. For example, in this study here, it was shown that if you inject a PEGylated protein into mice, then you have an anti-PEG IgM response. And then if you inject PEGylated protein for the second time, you have an association of the pre-existing antibodies with the injected protein. And you have binding of complement also, complement activation, and in the end uptake and clearance of the PEG antibody PEG complexes in the liver by the RAS. And you see that here, the pre-treated mice have a very different PEG interferon, PEG of albumin kinetics in the blood than those from those that have not been pre-treated. So we know today that PEGylated products are very common in the environment. They are present in cosmetics, they are present in shampoo, they are present in many of the environmental products that we are constantly exposed to. And other researchers study that, and about 60–70% of the general population has pre-existing antibodies. And this may affect the response to PEGylated drug products. So as we can see here, what we found was that 6 hours post-PEG interferon alpha injection, the patients without an NK cell response had much higher IgM anti-PEG in immune complexes than those with an NK cell response, suggesting that there's a stronger IgM antibody response that basically complexes the PEGylated interferon and then drives its clearance in the RAS by the Kupffer cells in the liver. And the same, yeah. And then you see the graph on the right. You see the same true for the 24-hour time point. So we were not able to see a difference in the general PEG antibody IgG or IgM levels in the blood. But as I mentioned, the whole process is basically the sum of the antibody production and the antibody clearance and the clearance of the complex PEGylated drug products. And so you have to look at the kinetics of all and the effects of all. So here we just see a difference in the IgM titer of anti-PEG antibodies that we quantitate in isolated immune complexes. But we also see a difference in the B cell response. So we differentiated B cells into antibody secreting cells from these patients and then measured the production of PEG antibodies, PEG IgM antibodies from these B cells, and then compared patients with and without NK cell response. And we saw differences, and we also saw a correlation between the strength of the PEG-specific IgM response in this ELI spot and the intensity of IgM staining in the liver histology. And again, in this correlation on the right, you see patients with and without NK cell response indicated by the different symbols. So it also differentiates between those two groups, suggesting strongly that a differential IgM PEG response, PEG-specific IgM response, correlates with clearance of PEG interferon alpha in the liver and with the induction of the NK cell response. So those patients who have a more rapid clearance of the PEG interferon alpha IgM complexes in the liver have less of an NK cell response. And we show this here in immunostaining in the liver. So you see patients with NK cell response and patients with NK cell response 6 hours after the first PEG interferon alpha injection in the collected liver biopsies. And you see in patients without NK cell response, PEG signals in the liver that basically co-localize with CD68, which is a marker of Kupffer cells and macrophages in the liver, and also localizes with IgM. So, and again, this separates the patients with and without an NK cell response, as shown by the different colors here in this correlation analysis. So it's difficult to explain with words. I noticed that when I spoke, but we try to make a graphical abstract here. So we think that in a patient population that is being treated with PEG interferon alpha, there are differences in the rapid kinetics of PEG interferon alpha accumulation in the blood. And you see that some patients have a much lower concentration of free PEG interferon alpha. And this is because they form PEG interferon alpha anti-PEG antibody complexes that then are cleared by liver sinusoidal cells, by Kupffer cells and other macrophages in the liver. And that basically determines how much PEG interferon alpha is left to activate NK cells and to drive ISG expression, and therefore determines the response to the injected PEG interferon alpha. And we suggest that this influences the overall treatment response. So it might be possible to improve the PEG interferon alpha pharmacokinetics and availability by changing this kinetics therapeutically. And also we have to be careful to think about the large market of PEGylated drugs that is ever expanding, and similar phenomena may also occur with other PEGylated drugs, which are used in much more chronic diseases such as cancer therapy and other chronic diseases where it's not as easy to have a fast readout a few hours after injection and to just look at ISG or NK cell responses. It's much more difficult to look at a treatment response against tumor cells, and this may also be influenced by the pharmacokinetics and availability of the injected PEG-related drug. So with this, I would like to close. I would like to acknowledge the people from my section on the left who contributed to this work. I mentioned most of them. So the HDB studies were all done by Hellenic Kefalakis with the help of Myung-Kyung Yang and Chyla Horgan, a postbac Erta student who is now in medical school. And Akira Nishio did the HBV studies in collaboration with Fabian Bolto, who started them here in the section, and with help from two students, Sharika Hassan and Nana Park, who are now also both in medical school. This would not have been possible without the clinical collaboration with my clinical collaborators, Margani, Theo Heller, and Chris Koh, and clinical fellows, Devika Kapuria, helped a lot with these HDB studies. We had help from Georgios Amanakis, a cardiologist who helped us with the FLITSNI analysis and the bioinformatics, and of course pathologist David Kleiner and Kazuyo Takeda from NCI and NHLBI, respectively, who helped with the immunostaining. And overall, I'd like to thank the branch, the liver disease branch, for this great environment that makes these unique translational studies possible. I would like to announce that we have an opening for a clinical scientist, a clinician, a staff clinician, who starts new studies. And if you're interested, please contact Jake Liang, our branch chief. It's open now, and we're just starting to consider applications. I also have postdoctoral fellow positions in my lab and student positions. So if you're interested in more the research side, the immunological side, please contact me as well. Thank you very much.
Thank you, Barbara. That's a very fascinating presentation. So now, can you see the question in the chat box?
Yes.
Yeah. So let's start the question from Tim. So in your presentation, you talk about after the interferon treatment, you see a spike of the HBV DNA in the blood. So what his question is that what's the resource that HBV DNA is integrate HBV DNA, or is some residual replicated intermediates from the cells?
Yeah, so this is a PCR result. So we think it's not replicating HBV DNA because the patients are on nucleoside analog treatment. So there's nothing that can replicate. So we think it's really released from infected hepatocytes, possibly from integrated DNA, but otherwise just HBV DNA from these hepatocytes, but not an increase in replication.
Okay, thank you. So next question is from the shipper Josh. So her or his question is, can baseline anti-PEG antibody inform the kinetics of response to PEG interferon alpha?
Yeah, that's what we had hoped for, because then you can identify patients who may benefit more from PEG interferon alpha therapy, but we did not see that for IgM and for IgG. So we think it's really the response that is triggered. So many patients have detectable antibodies prior to treatment, but then it's a kinetic of production and clearance after the injected PEG interferon alpha. And that's difficult to follow. So we were looking for a long time for a validation cohort. We have similar data on HCV patients that are included in the supplementary information in the paper, because there we also had stored samples 6 hours, 24 hours, 48 hours after the first PEG interferon injection. And we saw similar correlations confirming the results basically that we had in the HBV cohort. But we were not able to predict that by just looking at baseline antibody levels.
Yeah, thank you. So next question is from Dr. Balagopal. So first, he congratulates you. That's his fascinating work on the anti-PEG IgM and the interferon response. So his question is that any way to reconcile the HBV genotype specific difference in interferon responsiveness with the anti-PEG IgM antibody?
Yeah, we stratified. So in this very small study, most of the patients were genotype A infected. And so there was the two different subgroups did not differ in HBV genotype in this study. So I think the genotype adds a second differential response that needs to be considered differently. But I think this just depends on the genotype. Genotype A has a different response than other genotypes. But if you stratify by genotype, as in the study where most were genotype A, you still see a difference based on the anti-PEG response, the humoral anti-PEG response.
So next question is come from Dr. Li Shan Su. So his question is, have you looked at anti-HBs antibody in NK response and NK non-response patients?
No, we didn't. We didn't. Yeah, so it would be interesting to look, because presumably if you have S antigen in the blood which circulates in high levels, if you complex that, or if you have more HBs, anti-HBs complexes, that may also result in differential NK cell activation, I think. But I don't think that has been studied.
Okay. So next question from the Geoff. That's the question is, response to chronic HBV infection to the standard non-pegylated interferon is also variable, unrelated with antibody to the PEG.
Yeah.
Would you not also need to explain the differential response to interferon in such a patient?
I don't know. No, I really don't know.
So next question from that question is, at 24-week PEG interferon level are comparable. Any thoughts why NK cells do not respond at that time?
I'm sorry, I don't understand the question.
Yeah, I think that's let's say
24 weeks.
24 weeks of the treatments. That seems the pegylated level is already catched up. Why not?
Yes, so I try to explain this. So only after the first PEG interferon alpha injection did we have samples, biosamples, 6 hours and 24 hours after that injection. All of the subsequent samples were 1 week after the last injection. So it could either be an overall saturation over time, or it could be that it happens 1 week after the injection that there's no difference anymore. So that that in both patient groups reach a plateau at that point. So it's a really kinetic response. So what I would think is that early after the injection, within the first hours after injection, you see a differential NK cell response in the liver and in the blood that depends on the concentration of PEG interferon alpha that's available, on the bioavailability. But 1 week later, which you have at all of the subsequent blood draw time points, there's no difference anymore. So it depends on how fast the response is.
Yeah, I think from the failure, have 2 questions. That's one, I think you may already partially answered. That question is that if IgM clearance of the interferon determines the treatment response, so do you think when you extend the interferon treatment more than 48 weeks, does that going to increase cure rates or responder rates?
I would think no.
Yeah. So next question is that you characterize the NK response, the role of the NK cells in interferon treatment response. So do you know any role of the T and the B cells in the interferon treatment? Yeah.
Yeah, I think the B cells would be relevant because we're injecting interferon, so I don't think that T cells play a big role there. So the interferon activates the IgG response in the liver, which is important as an antiviral response, and it also activates cells like NK cells that kill hepatocytes. So these are also important effector cells and important readouts. But I don't think the T cell response is exhausted. There's a low frequency of virus-specific T cells. They express all kinds of exhaustion markers. They have no good effector function anymore in chronic infection. And also interferon is not the best way to activate these T cells. So I don't think a T cell response plays a role in the response to PEG interferon.
Yeah, next question is from the Kala Coffin. So her question is there a PEG interferon treatment calculator, such as incorporated surface antigen level and genotype, sex, viral load, etc., and interferon stopping rules? How would this be related with baseline anti-PEG antibody levels?
I don't know. So really don't know.
Yeah.
I mean, this is both studies that I presented basically result in new hypotheses or approaches. It would be great if people do interferon studies to get an early blood sample to see whether there are differences. To get a sample 24 hours after the first injection or 6 hours after the first injection and to see.
Yeah, this question from the Dr. Han, that's very specific. Have you seen whether intrahepatic HDV specific CD8 Tcells express CD103 as HBV specific Tcells does?
Sorry, can you repeat again? Whether intrahepatic HDV?
Yeah, HDV specific Tcells express CD103.
Yeah, I can't answer right now. I'm sorry, don't have the answer.
Yeah. So Dr. Jinheng Chang's question is HDV with escape mutation on the CD8 epitope replicates competently?
Whether HDV replicates if it has escape?
Yeah, when they carry that epitope escape mutation.
Yeah, I would think so.
Okay.
I would think so because because the patients are viremic, so they have escape mutations and they are highly viremic.
Yeah. So Dr. Hetago asked, does the anti-PEG IgM boost interferon response in macrophage and dendritic cells?
Sorry, what boosts? Can you repeat again?
Yeah, I think that's underlying consideration, I guess, is that because the IgM pegylated interferon complex was uptake by dendritic cells and macrophage.
Mm-hmm.
So his question is that does that actually boost the interferon response in those cells?
I also don't know. I would think that it gets eliminated if it by the RAS, by the Kupffer cells, that this results in elimination. Whether it boosts the interferon response in the liver. Yeah, so, I didn't have any data on that.
Yeah. So another short question from Dr. Li Shansu is, does those liver-resonant T cells also express TIM3?
I don't know. I would think so.
Yeah, there's two questions left. One is, does pegylated interferon sequestered in liver have no effects on hepatocytes or liver-resonant lymphocytes? Right.
Sorry, I didn't understand. Again,
I
need to read at the same time, but I don't find the questions fast enough.
Yeah. So I think that the time is already over almost 10 minutes. So I think that I must cut here.
Mm-hmm.
Yeah. So thank you, Barbara, and also thank you every attendees that for your question. And if anyone wants to, because at 12:15, that we will have a Bloomberg faculty meeting, a Zoom meeting with Barbara. So if anyone interested to attending, that you can send an email to either Tim or myself, so I can send you the link.
Mm-hmm.
Thank you very much. Thank you.
Yeah, and please apply for the staff clinician position.
Yeah. Oh, yeah.
Want to advertise again? Contact Jake Liang.
Okay. Okay. Yeah. Yeah. See you a moment later. Bye-bye.
Thank you.